Thank you very much for the organizers for this opportunity to share my research with you today. So the goal of this project was to figure out how we can move beyond the current resolution limits of single-cell epigenomic studies in an effort to really study gene regulation at the level of individual chromatin fibers within diploid organisms. So this is sort of a figure which you guys are probably used to seeing, but this is really indicating the primary architecture of gene regulation, which occurs at the level of individual chromatin fibers, as was really nicely pointed out in the last couple of talks, where diverse chromatin proteins such as nucleosomes, transcription factors, or polymerases occupy and actuate different parts of the human genome. This architecture is foundational for genome function, yet our current understanding of how neighboring features along the same chromatin fiber will work together still remains quite limited. So current methods mainly that are used for mapping chromatin architecture, such as ATAC-seq or DNase-1 or MNase-seq, really utilize these non-specific DNA editing enzymes to selectively cleave DNA at accessible regions of the chromatin, such as actuated regulatory elements or internucleosomal regions, and then short double-stranded DNA fragments are released from the genome and are isolated and sequenced using short-read sequencers to identify regions of chromatin accessibility, transcription factor binding, or nucleosome occupancy. These approaches have recently been adapted, as we heard about throughout this conference, to enable the chromatin architectures of individual cells. And this process has really been instrumental over the last couple of years in revealing regulatory architectures of rare cell populations, as well as identifying elements that appear to co-actuate within individual cells. However, these single-cell methods are not without their challenges, and I think chief among them is the inherent sparsity of data that is collected from each individual cell. For example, even with the relatively high coverage single-cell ATAC-seq data set, you're really only getting less than 100,000 reads from each individual cell. And given the fact this is a diploid organism as well, most of those reads do not map to individual SNPs. So you really only know a piece of chromatin architecture of 1 in every 50,000 bases within that cells. And often to overcome this, you're aggregating hundreds, if not thousands of cells together to identify chromatin features or transcription factor occupancy events. And this is further limited, as I mentioned, when it comes to diploid cells, where you actually don't just have one genome, but actually have two genomes that you need to interrogate that are not necessarily the same. So to overcome these challenges, we sought to construct a system for stenciling the architecture of individual chromatin fibers onto their underlying DNA templates in a manner that enables them to be sequenced at base pair resolution along multi-kilobase fibers. And to accomplish this, we've leveraged several prior observations in the field, some of which were nicely talked about by Aaron earlier, which is that a sort of sequence-specific adenine methyltransferase DAM or sequence-specific as GPC or CpG methyltransferases can be used to selectively mark sites of protein occupancy along the genome. However, we wanted to build upon these methods such that we could achieve near single nucleotide resolution and could map chromatin fibers of over 10 kb in length. And to do that, we sought to evaluate a novel class of enzymes called non-specific adenine methyltransferases. And we chose this class of enzymes for several important reasons. First, AT bases represent over half of the base pairs in the human genome, giving this approach a near base pair resolution. Also, M6A is essentially a non-native DNA modification in all eukaryotes, so we don't have to worry about seeing this signal endogenously. And finally, M6A methylation does not alter the underlying genetic code and does not fragment chromatin fibers, unlike the enzymes used in prior approaches. So as an initial proof of principle, we isolated purified non-specific M6-MTAs HEA5 and treated nuclei with this and just a limited amount of this enzyme, and followed by DNA extraction. And for this initial experiment, we fragmented the DNA into short reads, immunoprecipitated the M6A modified DNA using an antibody, and sequenced these immunoprecipitated fragments using an Illumina sequencer. So as is shown here, this method sort of demonstrated that M6A MTAses can selectively mark sites of chromatin accessibility as recapitulated by using DNase-1-seq as sort of the gold standard. First of all, just to evaluate whether or not this was a general property of this class of enzymes, we isolated 4 additional M6A MTAses and demonstrated that all 5 of these enzymes were similarly able to selectively mark sites of chromatin accessibility. Okay, so after demonstrating that these enzymes actually can work as a probe of chromatin accessibility, we next wanted to determine whether or not we could use this class of enzymes to generate single molecule chromatin architectures, which was our goal of the study. So of note, for the next set of experiments, we actually increased the amount of M6A MTAs by over 100-fold to really achieve single nucleotide or near single nucleotide resolution. And to generate these highly accurate sequencing data on these fibers, we utilize the PacBio Hi-Fi sequencing, as circular consensus reads enable really high base pair accuracy. And importantly, actually, unlike nanopore sequencing, since PacBio sequences both strands of the read, it actually doubles our resolution and avoids the issue of potentially duplicate fibers in our output. So in addition, PacBio sequencing enables us to directly read out which of these bases are actually modified as an M6A, as once the polymerase actually hits one of these M6A modified bases, it causes the polymerase to temporarily pause, which can be directly read and identified using the raw sequencing data as is shown here. So here I'm displaying a representative human locus sequence using FiberSeq. And to orient you to this data, the gray line here represents an individual chromatin fiber that was sequenced, and the purple dashes signify individual sites of M6A modified bases, denoting bases that are accessible to M6A MTA's activity and hence not bound by a protein. So using these single molecule patterns of M6A modified bases, you can readily identify patches of actuated regulatory DNA, as well as patches that appear to be bound by a protein because they're occluded from methylation by this enzyme. You can also compare these fibers across, or compare these patterns across multiple fibers to construct the actual chromatin architecture of individual fibers, and really appreciate the elements that are homogeneously versus heterogeneously present across a population of cell. So furthermore, by comparing this architecture of dozens of fibers overlapping a given locus, you can really appreciate the widespread heterogeneity that exists in the primary architecture of individual chromatin fibers across a population of cells. So with some elements only active within a select population of cells, and other ones like these here showing over twofold difference in the actual DNA content of the actuate element across individual fibers, significantly altering sort of what we think of as actually the accessible DNA within a given regulatory element that the individual cell is appreciating. So these mapped chromatin fibers are typically over 10 kb in length, which enables us to determine the principles guiding the coactuation of neighboring elements along the same fiber, such as these two fiber or elements that are located over 4 kb apart. So when we do this, one of the largest determinants we see of neighboring elements being coactuated on the same fiber appears to be how far apart they are, with closely spaced elements such as these two, being coactuated on the same fiber in 75% of the case, versus more distantly spaced elements being coactuated less than 50% of the time. Moving into individual regulatory elements also enables us to evaluate single molecule transcription factor binding events, as is seen with this CTCF binding element, where only 2 of the 4 chromatin fibers overlapping this location actually appear to be bound by CTCF. And overall, this approach exposes that around a third of the fibers overlapping a ChIP-seq peak actually contain a nucleosome at that position, indicative of a closed chromatin state, whereas only 20% of the fibers actually do contain CTCF occupancy. And notably, CTCF binding elements that are contained, these chromatin fibers that are preferentially bound by CTCF, were more likely to be maintained within long-range chromatin interactions, providing further sort of single molecule validation to the loop exclusion model that CTCF occupancy at a given site is able to restrict cohesion movement along that fiber as it passes by. So in the last minute or two, I just want to present some data showing how you can combine these single molecule chromatin architectures with underlying genetic data to actually shed light on the function of the non-coding genetic variants. And for this experiment, we subjected primary human CD4 cells to the FiberSeq. I just want to highlight here that FiberSeq is nearly identical to standard high molecular weight genomic DNA extraction methods, with the exception of 2 additional steps, which together take only 25 minutes to perform. And the level of sequencing for this particular sample was sufficient to really understand what's going on, and we really get high base pair accuracy, which is sort of the standard right now with PacBio SQL 2. Importantly, these reads are actually quite long, and this is really helpful because what that actually means is that over 80% of the reads end up overlapping a heterozygote variant, enabling us to phase nearly the entire epigenome within the sample. So here's just two examples that I just want to go over. And as you can see here is that if you just do standard sequencing, you would just identify this heterozygote variant observed in sort of the PacBio Hi-Fi reads. And by simply overlaying the epigenetic information that we're collecting from the same reads, you can readily appreciate that the reads fibers containing the A allele are preferentially inactivated, whereas the fibers containing the G allele are preferentially activated, a finding that we can subsequently confirm using DNase 1 data as sort of the gold standard here. This is another example here where you can similarly see that the fibers containing the T allele are preferentially active. And overall, this process is able to identify variants in the cell associated with altered chromatin architectures, really providing a direct readout for the functional state of the non-coding genetic variants with long-read sequencing. So overall, I think that this sort of method really helps us to better understand how chromatin fiber architecture is existing and the heterogeneity that actually exists at this level of gene regulation. And similarly, it can be utilized to better understand how non-coding genetic variants actually impact their overlying epigenomic architecture. And with that, I would like to thank all the members of my lab, as well as Brian Debo and Eric Haugen, who have really been instrumental in this project. And also, I'd like to thank the generous funders for this, their support, and all you for your attention. Thank you.
All right, that was really fantastic. So I'm going to kick us off with a couple of questions. And the chat is open. I was wondering if you were to, let's say you were to probe a cell type that was heterogeneous in terms of accessibility, using FibroSeq, and if you were to plot like a histogram of a particular chromosome that was variable from cell to cell because of cell cycle or because of populations in terms of accessibility on the single molecule level, would that histogram sort of recapitulate what you might expect to see in single cell ATAC-seq or something like that? Do you expect there to be concordance, or is it too noisy and sparse in either of the other measurements to compare?
That's a good question. I've not done the exact experiment which you're asking for, so I can't sort of say definitively, but I would expect that once you start to actually aggregate multiple cells together in single cell ATAC-seq data, that you're going to start to approach what we're seeing here with the per chromosome level. And we've seen this as well, that if we actually compare and aggregate our per chromosome or per molecule data together at a given locus and compare it just to bulk ATAC-seq or DNase 1-seq measurements, we get R squareds of around 0.8 or better. So yeah. Which is really, we start to sort of, this bulk stuff are really, giving us a bulk picture of what's going on.
But I guess another way of asking the same question is, we know that like single cell ATAC-seq is extremely sparse because, the efficiency of TN5, tegmenting these linkers is low, and then we have to amplify and so forth. So how would you compare the efficiency or the sensitivity in this approach? Do you between methyl A calling and just the efficiency of methylation on the single molecule level, is it pretty dense? Like when you add all that MTAs into one experiment?
Yeah, that's a very good question. So we've actually done this experiment. So what we've essentially done, where we've just titrated the amount of M6A MTAs we're getting, because really we wanted to max out the amount of M6A MTAs to really get as good of resolution. So we did a titration from 0.2 units to 2 units to 20 to 200 to 2,000. And essentially what we saw is that once we go above around 50 units of M6A MTAs, we're essentially maxing out, and we're not really improving our resolution. And right now,
and do you start to get background, or are you just not improving resolution?
You're not really improving the resolution that much. So what you can see is that in activated elements, you are seeing roughly, I'd say, every other, maybe a little bit more than that, of the adenines that could be methylated, actually methylated at these levels. And there's is some sequence specificity for all these M6A MTAs are not perfect, so they don't methylate all adenines equally, but really beyond just poly A stretches, we're getting really good methylation within them. I'd say in terms of your question, in terms of background, the thing that we do start to see, and this is actually something which we're actively looking into, is that you can actually start to measure processes that are dynamic during this reaction. So this reaction is going for 10 minutes. And you can imagine that if you're actually adding more M6A MTAs and you have more of that available, you're theoretically able to capture states within that 10-minute, which actually are quite rare. And so you can start to look at, say, a nucleosome unwrapping and unfolding and really capturing sort of more rare states within that process by adding.
Yeah, I was thinking about that in the context of DAM-ID. Like if you just had a cells that was expressing your HEA5, for example, or ECOG2 or something by itself, if you would get, if you get more measurements of more of that sort of chromatin reorganization as you're letting the cells sort of do their thing before extracting their nuclei.
Yeah, and I can say definitively that the answer to that is yes. We've done that experiment. I mean, the nice thing, as you kind of point out in your talk, is that M6A, as far as we can tell, is really not toxic to these cells, or at least I've only done this really with transformed cell lines. But really expressing this at high levels, massively methylating the genome, is really not causing a significant impact in terms of the cell function, at least measured based on bulk RNA-seq or chromatin accessibility measurements. So.
Great. Okay. Do we have any other questions in the chat? Okay. Thank you so much, Andrew.